Yeah, good afternoon. So we will talk about outcomes in we were talking about outcomes in case control study. Uh, right. We were talking about outcomes in a case control study. A case control study provides the direct estimation of the exposure rates to the suspected factor in disease and non-disease groups. Okay. So for example, there are so if we construct 2 by 2 table, there will be in one column there will be cases, the other column there will be controls, and in the rows, one row there will be smokers, then there will be non-smokers. So as you understand, this A plus C is equal to the total number of cases, and B plus D is equal to total number of controls. The exposure rate in cases will be calculated as A by A plus B. Means the people who got exposed here, that means those who were smokers and developed some disease, that will be divided by the total number of people in cases. That will give rise to percentage of cases. Okay. Right. And if you want to see controls, the controls will be B. That means those people who got exposed to smoking but did not develop did not develop any disease, that will be 55 B divided by total number of controls. So that will give rise to percentage of total number of controls. That is how we calculate exposure rate. Now coming to odds ratio and relative risk, relative odds. The odds means odds of an event is defined as the ratio of the number of ways an event can occur to the number of ways an event cannot occur. So it can be calculated as if the probability of event X occurring is P, then odds of it will be odds of occurring is P divided by 1 minus P. Odds ratio of the odds that are the cases were exposed to the odds that the controls were exposed. Okay, the ratio of odds that the cases were exposed to the odds that our controls were exposed. Odds ratio continuing odds ratio using the fourfold table. Now here we are taking a fourfold table. Using a fourfold table, the odds ratio will be odds that cases will exposed, odds that case will exposed divided by odds that control will expose. Okay, odds of that cases will expose and odds that control was exposed means odds ratio. So that the formula will be like A A divided by C whole divided by B divided by D. Okay. I hope you understand this. I will try to repeat again. The odds ratio and relative ratio means the odds of event is defined as a ratio of the number of ways an event can occur to the number of ways an event cannot occur. If the probability of event X occurring is P, then odds of it occurring is P divided by 1 minus P. Ratio of the odds that the cases were exposed to the odds that the controls were exposed to. So it is a ratio between two odds, cases odds and control odds. So the formula goes like odds that cases was exposed, odds that controls was exposed. So A divided by C whole divided by B divided by D, which is equal to AD by BC. Right. Odds ratio, which is also called as cross product ratio, can also be viewed as the ratio of the product of two cells that support the hypothesis of association. That means cells A and D, deceased people who were exposed and non-deceased people who were not exposed, to the product of the total cells which negate the hypothesis of association. When is the odds ratio a good estimate for relative risk in the population? If the cases studied are representative, that means regarding history of the exposure of all people with the disease in the population from which cases are drawn. So if the study are case studied are representative cases. In terms of control studied are representative of that area. Regarding the history of exposure of all the people without the disease in the population from which cases are drawn, right? When the disease is being studied, it does not occur frequently. Now, disadvantages or limitations of cross-sectional case control study are: there are susceptible biases if not carefully designed. The study especially susceptible to exposure and misclassification, especially susceptible to recall bias, restricted to single outcome, in incidence rates not usually calculated, and cannot assess effects of matching variables. Now, in terms of advantages, the only realistic study designed for uncovering etiology in rare diseases. It is the only realistic study designed for uncovering the etiology of rare diseases. It is important in understanding new diseases. It is important in understanding new diseases. It's commonly used in outbreaks investigation. It is commonly used in outbreaks investigations. Useful in inducing period is long. Useful if inducing period is long, and relatively inexpensive. Okay. So these are the advantages of case control study. Applications of case control study are: when there is a rare diseases, the case control approach are the most efficient for rare diseases. For example, idiopathic pulmonary fibrosis or most cancers. Cohort approaches would require large population and prohibitive expense and follow-up. Case ascertainment system in place. If the case so if the case ascertainment system is in place, the conduct of the case control study may be facilitated by the availability of a case ascertainment system like population-based cancer registry, hospital-based surveillance system, mandated disease reporting system. When funding and time constraints are not compatible with a cohort study. Okay, so these are the points where you can apply case control study. And now coming to nested case control study. What is nested case control study? We choose study population, and after study population we choose study population, obtain interviews, blood, urine samples, etc. Then we divide them into developed and developed disease, do not develop diseases, and then we follow them up as cases and controls. It is also a case control study, but it is a nested case control study, a little different design. Now nested case control study, okay. So advantages of nested case control studies: possibility of recall bias is eliminated since data on exposure are obtained before diseases are developed. Now exposure data are more likely to represent uh, the pre-illness state since they are obtained years before clinical illness is diagnosed. Third, the cost and reduction comparison. Cost are reduced compared to those of a prospective study, since laboratory tests need to be done only on specimen from subjects who are later chosen as case or as control. Important findings of case control studies are like, okay, so these are a couple of important findings like cigarette smoking and lung cancer and diethyl stilbestrol and vaginal adenocarcinoma and all those things. So there are some. Okay, so we are done with case control study. We'll start cross-sectional study. Without wasting time, I'll just start it and then we can continue in our next class. So in cross-sectional study, what do you understand by cross-sectional study? The cross-sectional study is cross-section, and there is a cross-section. What is the cross-section? We are cross-secting something. What are we cross-secting? We are cross-secting time. We are cross-secting time. So when we are cross-secting time means we are this is if this is the running time line. We are cross-secting at 2 points, and in between these 2 points, whatever we are trying to observe will be a cross-sectional study. It's just like a what is happening at this point of time. Okay, we are not looking retrospectively, we are not looking prospectively. What we are trying to freeze the time, and we are trying to see what is happening in this particular frozen time. Okay, so history and classification differences between, so we will discuss about history and classification differences between descriptive and analytical studies, attributes, advantages, disadvantages, case scenario, and guidelines. Then again, history, development of modern epidemiology, scope of clinical epidemiology, definition, need. So there are a whole lot of things here, but we will only focus on our subject because as an exam point of view. So why should a medical student know about epidemiology? Okay, so we are just I'll just skip this. Okay, now this classification, it's important. This classification, kindly write it down if you have book or something. It is always easy, always good to have. Uh, it's always good to have this thing. Okay, so in classification, the epidemiological design, there is epidemiological design, and there are different types of studies. They are mostly divided into two groups: observational studies or experimental or interventional studies. So any experiment you do, it's like an intervention. You are intervening in normal course of disease or normal course of time. Okay, so looking at epidemiological studies, epidemiological study, it's experimental and non-observational. In observational studies, specifically, you further divide it into analytical and descriptive studies. And analytical study is again subdivided into three types of studies: case control, cross-sectional, and cohort. So case control study, cross-sectional study, and cohort study are analytical study. Now in observational study, descriptive, it's longitudinal, cross-sectional, and etiological studies. In epidemiological studies, in the experimental studies, it's divided into three groups: clinical trials, field trials, or community intervention trials. Okay, randomized control trial specifically. Okay, according to hierarchy of the scientific evidence, randomized control trial are the most confirmatory trial around. So first you can see that how the studies are developed. First there are case reports which comes out, then there are case studies which comes out, then people do cross-sectional study to look what is happening, then they make out the hypothesis, then this next step is case control study, the next step is cohort study, and then next step is non-randomized control trial, sorry, non-randomized trials, and then randomized trials. So cross-sectional study is studies entail collection of data, the cross-section of a population, and comprise of whole population of or a whole population or a sample of the whole population. Cross-sectional studies otherwise are called as prevalence studies. So prevalence rate is equal to number of prevailing cases of a disease divided by exciting at each given point of time, further divided by uh, estimated population at the same point of time, that particular time, into 1000. Now observational study, it's a single examination of population at one point of time. Individual individual based and measure exposure and treatment. So measure exposure and effect. Exposure exposure proceeds for following the effect are not known. So, sorry about that. Feeling very tired with this mask. Just take a fresh breath and okay. So the observational study is a single examination of the population at one point of time. The individual it is individual based and it is a measure of exposure and effect. The exposure precedes or follows the effect, which is not known. So descriptive cross-sectional study or population survey or prevalence survey, it's also known as these three terms interchangeably we use. The information about single or multiple variables can be obtained and estimate problems and or prevalence can be and or prevalence of the problems can be obtained. Point prevalence, it's divided into point prevalence, period prevalence. No, it can be of three types: point prevalence, period prevalence, or disease and suspected risk factor population or specific individual. The analytical cross-sectional study is the information presence and strength of association and the testing of hypothesis can be done. By definition, an analytical investigation in which subject are sampled at a fixed point or period of time, and then the association between the concurrent presence or absence of risk factors and disease are investigated. Now, descriptive cross-sectional studies are it describes the presence of the disease, disability, and symptoms of ill health, dimensions of positive health such as fitness, attributes to related health body measures and blood pressures. Analytical cross-sectional study, strength of association between disease and risk factors that shows strength. So in analytical cross-sectional study, we explore strength of association between disease and risk factor, determinants of the disease or conditions, and predictors of disease or conditions. Attributes of the cross-sectional studies are: population studied comprise of survivors that at a point or period of time. attrition may have occurred because before the study. It describes association between exposure and disease simultaneously. It measures prevalence of disease in exposed and unexposed, risk measurements and prevalence ratios and odds ratios, and evidence of causality in is only suggested. It is more prone to selection bias. The design of cross-sectional study is usually like sampling, reference population, and then sampling. Okay, so we take reference population and we draw sample out of reference population, and in between we do sampling. Okay, sampling means defining the population and measuring the sample size and defining the sample. The characteristic, okay, the sample means, so okay, so the sample can have the different characteristics. The characteristic, that is exposure and outcome, characteristics and disease, then characteristics and again no disease, no characteristics and disease, or no characteristics or no disease. So these are the four circumstances which a sample can have as a property. So talking about exposure and disease measures simultaneously are time. Time is a key. The present disease and exposure. The present exposure is future and disease, and the case control study will define present disease and exposure. So cross-sectional study covers cohort and case control study as well. Cohort study becomes more specific and case control study becomes more specific than cross-sectional. So I'll stop here for now. On 20th slide, we will discuss about steps in conducting cross-sectional studies in our next class. Thank you. Have a good day.